it's my great pleasure to introduce today's speaker, Dr. Sarah Hainer. Sarah conducted her doctoral studies at the University of Pittsburgh in the lab of Joe Martins, where she did fantastic work on chromatin-mediated regulation of transcription in budding yeast. She then did her postdoc with Tom Fazio at the University of Massachusetts, studying the role of the BAF complex in mouse early development. In her work, she also developed single-cell cut-and-run and used this to look at the function of BAF regulating pluripotency factor binding in early embryos. In 2018, she started her lab at the University of Pittsburgh in the Department of Biological Sciences and continued studying the role of chromatin remodeling and transcriptional regulation. As well as being a great scientist, Sarah is also a strong advocate for underrepresented minorities and has been part of a number of initiatives to promote inclusivity in science. And she also won a number of awards, and I won't list them all, but the most recent award listed on her department bio does stand out to me, and this is the 2020 Most Valuable Professor Award given by the University of Pittsburgh Women's Diving Team, which I think is just great. If you want to hear more about her diving prowess, I suggest you stay for the meet and greet session after the seminar. Anyway, I'm very excited to hear from Dr. Hainer today as she tells us about her work profiling pluripotency factors in single cells and early embryos. So please join me in giving a very warm, if socially distanced, welcome to Dr. Sarah Hainer.
Thank you, Ben, for that introduction, and thank you for hosting me today. So I actually did want to start off a little bit unconventionally and spend the first 2 minutes to just remind everyone that there's a mountain of data demonstrating that women, disabled academics, Black academics, non-native English speakers, people from specific geographical locations, and more are discriminated against in academics in aspects ranging from hiring to obtaining grants, to salaries, to paper citations, and more. All the things required to remain active researchers. But there is also data to show how bad this is for science. Oopsie. Diversity breeds innovation. Innovation drives science. Hindering inclusion of diverse perspectives only hinders science. Many of these issues seem to be beyond any one individual's capacity to correct. And while it is true that there is a huge systemic problem that perpetuates these issues, I want to remind everyone that each one of us has the ability to make change. That's all of us, from faculty to trainee, from junior to the most senior of scientists. We all need to identify and combat our own biases. We can all change how we interact with and support one another in our lab, department, institution, scientific field. We can change how we mentor, review grants, review manuscripts, hire people, write and read recommendation letters, and more. Science has a systemic issue, but science is made up of scientists, and we are the ones who have to break through these issues. To me, a community like this is a starting place to do so. This community was built to support one another in a really tough time. So let's continue to show that support for individuals who are marginalized within academia, and let's work together to make more changes in our local and scientific communities. So with that, I'd like to thank the fabulous group of organizers for putting this whole community together, and of course for hosting me here today. So I'll leave my initiatives there and start talking about some science. Our lab is interested in self-fate. That is how a cell retains its own identity, or in the case of a pluripotent cell, such as the one shown here, how it differentiates to become one of many potential cell types. Of course, this process and subsequent developmental processes are what permit the development of our favorite multicellular organisms. An improper cell specification or dysregulation of these processes, of course, results in disease states. So more specifically, our interests are in how this one pluripotent cell can give rise to the differentiated cells, to any of these differentiated cells. Of course, this is largely due to regulated gene expression. In a liver cell, you have to turn on alcohol dehydrogenase genes, and in a neuron, you have to turn on neurotransmitter genes. But in the opposite cell type, those genes are off. So really, our main interest is how different sets of genes are expressed in different cell types during development. But since every cell has the same DNA sequence, what molecular mechanisms contribute to transcription regulation to provide differential expression patterns? And it isn't a surprise to this audience about the mechanisms we think about involving transcription factors, chromatin organization, and non-coding RNAs. You all know that transcription factors bind to specific DNA sequences at regulatory regions and can modulate transcription. Of course, at the heart of all DNA templated processes in all of our hearts is the stable association of DNA with histone proteins, where accessing DNA regulatory elements is established and maintained by these transcription factor binding and chromatin regulators like remodelers that function together to drive cell type specific gene expression. And finally, a more recently uncovered regulatory mechanism is the action of non-coding RNAs. And while our lab's interested in all three of these molecular mechanisms driving cell fate and gene expression profiles in specification, today I'm just going to focus on the first two. Oopsie, sorry. So how are different sets of genes expressed in different cell types during development? You all know that different cell type specific factors are expressed that can drive expression of these cell type specific genes. For example, in blastocysts and in the cells derived from the inner cell of the blastocyst or embryonic stem cells, OCT4, SOX2, and Nanog are important transcription factors required for pluripotency of these cells. You also all know and have heard a lot through this series that nucleosome remodeling factors are important for appropriate gene expression. Summarized here are what nucleosome remodelers are required during different stages of embryogenesis and essential for the normal development of embryos. And you can see that mammalian switch SNF or BAF is essential throughout development and tissue specification. And for a long time, I've wanted to answer the question of whether nucleosome remodelers regulate pluripotency factor binding in blastocysts. For example, the BAF complex is expressed prior to blastocyst formation, and therefore it would make a lot of sense to me that this remodeler would regulate the localization of OCT4, SOX2, Nanog, these pluripotency factors that are expressed in the blastocyst. So just to divert a little bit and quickly introduce BAF, because it's a main player here, the BAF complex is highly conserved, a nucleosome remodeling complex that has a specific assembly of subunits in embryonic stem cells called embryonic stem cell specific BAF or ES-BAF. And of course, the sole catalytic activity of BAF is to translocate DNA to result in nucleosome sliding. And there's only a single catalytic subunit in ES-BAF, whereas other BAFs actually contain two catalytic subunits, and that's called Brg1. I told you that BAF is essential during embryonic development, and in fact, homozygote knockout mice die during the peri-implantation stage, so before day E3.5. In embryonic stem cells, depletion of Brg1 results in a self-renewal defect, as observed by decreased alkaline phosphatase staining, which is a marker of stem cells. And finally, down here, I'm showing you some cancer genome sequencing efforts that revealed an unexpectedly high rate of mutation of BAF complex genes, which are collectively mutated in about 20% of all human cancers, which is huge. Okay, so returning to this idea that BAF may regulate pluripotency factor localization in blastocysts, previously we published data from mirroring embryonic stem cells analyzing small footprint reads from mRNA sequencing data. And these data suggested a modest role for BAF in regulating OCT4 and SOX2 occupancy. So when the ATPase Brg1 was depleted in comparison to the control GFP depletion, we saw a modest reduction in small mRNAs footprinting, or transcription factor binding can be inferred from that relative to that control. Further and more elegant work from Robert Close's group showed that upon tamoxifen induction to reduce Brg1, there was a decrease in OCT4, SOX2, and Nanog occupancy by ChIP-seq at only a subset of the binding sites. And so these results are still surprising to me because of the developmental expression profile I showed you earlier. So we really wanted to assess the role of BAF in regulating OCT4 SOX2 and Nanog occupancy in the actual embryo instead of the cells derived from the embryo. But to do this, we needed to be able to perform single embryo occupancy studies, which was impossible, of course. And that's because CHIP, the most widely used localization study assay, really requires a huge number of cells. So many of us are familiar with chromatin immunoprecipitation, so I'm not going to walk you through this technique. But CHIP is widely used to look at factor binding throughout the genome, and it's a super powerful technology, but typical experiments require between 1 million to 5 million cells, for example, and blastocysts are only composed of about 30 to 50 cells. But in recent years, many people have developed chromatin profiling methods based off of CHIP assays using a lot less cells. However, even the most sensitive of these assays, like CHIP mutation and carrier ChIP-seq for transcription factors, were limited to only about 10,000 cells for transcription factors. I'll note that for histone modifications, people were able to apply these technologies to much lower cell numbers. And I'm sure this group doesn't need to hear this, but just for a quick reminder, histone proteins are far more abundant. So it's a lot easier to profile histones than it is for these transcription factors. So what technique could we use to really look at factor localization in low cells? Well, it's not going to be a surprise for this group that we turn to CUT&RUN, which is the genome-wide derivative of Uli Lamely's Sheik technology that Steve Henikoff's group published in 2017. So I know we've discussed CUT&RUN a lot in this group. I do want to quickly detail the method just so everyone's on the same page and because it's important. So in the way we do CUT&RUN, which is based off of the original Henikoff method, you release the nuclei by putting them into a hypotonic buffer, bind these nuclei to lectin-coated concanavalin A beads, diffuse primary antibody in, and then the key to Sheik or CUT&RUN is really the recombinant protein A emanase, where the protein A will recognize the IgG backbone of the antibody. And then you can initiate specific cleavage of DNA flanking the protein of interest by introducing calcium. You chelate the reaction through addition of EDTA and EGTA, and then release these soluble fragments through centrifugation. Then you can build Illumina libraries and prepare them for sequencing. And so for anyone that hasn't tried CUT&RUN yet, I highly recommend it. It's about 3 times faster than CHIP. It provides much higher signal and lower background. And as someone who did 1 million CHIP assays in their PhD and postdoc, I can't recommend it enough. Okay, so we slightly altered the conditions that the Henikoff group did for CUT&RUN and applied it to embryonic stem cells from 500,000 down to 10 cells. And that's what I'm showing you here. This is relative occupancy of CTCF. We chose CTCF because it has a really great epitope. The antibody is amazing. It binds to 30,000 locations in the ESL genome, for example, and it has a really good consensus motif. So we can do great analysis to make sure the assay is working. So these data are shown over previously established CTCF sites, and all the data I'm going to show you in heat map form are shown that way. So these are CTCF sites that were described from CHIP assays, and these are just sorted by the strength and the CHIP peak. And up top, you have the CUT&RUN for CTCF, and below you have the no primary antibody control. Each row here is a different locus for an individual CTCF binding site. And hopefully you can appreciate that even down to 10 cells, we were able to get robust profiling of CTCF relative to the no primary antibody control. And so just to show you these data in a different way, these are just meta plots over the same location. So this is just an averaging of those data. And you can see the nice enrichment for all these. And then if you look at the 2 different replicates, And if we take the ChIP-seq data as really the gold standard, where ChIP-seq represents 100% of the peaks identified, we're really capturing about 90% of CTCF ChIP-seq peaks down to 50 cells, and then even 10 cells we're capturing a good number of the previously established peaks. I do want to point out that you would never have expected to obtain 100%, because there are probably false positives in the ChIP-seq data, and also these are two totally different techniques. But the fact that we're recovering about 90% is pretty remarkable. And then here is just a simple genome browser track at a specific location. You can see down to 10 cells, we get a nice peak compared to the ChIP-seq published data. And there is this one location right just adjacent in the ChIP data that isn't found in the CUT&RUN data set, which suggests that this is either a false positive in the ChIP or it's unique and cannot be identified by CUT&RUN. And so there are those peaks, and there's also peaks the other side, which are identified in CUT&RUN and not in the ChIP assay, that are interesting to think about. So I guess these are demonstrating pretty sensitive mapping by CUT&RUN. And so we also assayed, I don't want to belabor this because it's all published, but we assayed histone H3 lysine 4 trimethylation in similar cell numbers and saw robust profiling as well. And then looked at a couple other histone modifications because CTCF and K4 trimethyl have been proven to be among the most robust epitopes for ChIP-based studies, which raises the question about whether CUT&RUN could really effectively map a broader array of histone modifications in DNA binding proteins. To really explore the utility of CUT&RUN for mapping these diverse factors like histone modifications, we generated 50,000 and 50 cell profiles against histone H3 lysine 27 acetylation or trimethylation, again observing robust enrichment. But then turning back to assaying these pluripotency factors that we want to eventually examine in the embryo, we again saw robust enrichment over previously established ChIP peaks for these factors. And so we can build really robust libraries using CUT&RUN to profile transcription factors from as low as 10 cells for CTCF or 50 cells for these pluripotency factors shown here. Again, I threw these in just because they told me I had extra time today. I don't really want to belabor the point, but here's the peaks for the metaplots for OCT4, SOX2, and Nanog showing robust binding over the no primary antibody control. And I think, again, what's exciting is that we're able to identify the previously established ChIP-seq peaks to pretty high reproducibility between the different replicates and even in low cell numbers. So the data that I've been showing you thus far has really been over previously established ChIP-seq peaks. But you can also, of course, call peaks from these 50,000 cell or 50 cell data sets and perform motif enrichment analysis to identify what motifs are found underlying those peaks. And so this would just be peak calling followed by motif analysis to identify which motifs are there. So this is not dependent on any previously established data other than the motif information we need to have. And what I hope you can observe is that even in the 50 cell, we get motifs that make a lot of sense. If you zoom in, if you focus in on these SOX2 binding sites, we're getting the SOX factors enriched, their motifs enriched, which is very nice. So now we can turn to the question I wanted to ask for such a long time, which was, with application to such low cell numbers, can we transfer this technique to individual embryos? And so we paired up with a really talented embryologist, Anna Boscovich, and with Anna's help, we obtained blastocysts and applied CUT&RUN for CTCF on these single embryos. And what I hope you can appreciate here is that we did get nice enrichment for CTCF in individual embryos. And importantly, these data are actually visualized over embryonic stem cell sites for CTCF because, of course, we don't have any previous data for CTCF binding sites in blastocysts. And so we are now finally poised to ask our really important question of whether the nucleosome remodeling complex BAF is necessary for pluripotency factor binding in vivo. And so just as a really quick reminder, BAF is expressed early in embryogenesis. And the question I want to ask is whether BAF is required for localization of these pluripotency factors expressed in the blastocyst, specifically focusing on Nanog first. And so to perform this assay, Anna injected zygotes with siRNAs that we had previously validated. And these were targeting SMARCA4, which is the gene encoding the ATPase for BAF, or Nanog as a control. We grew these zygotes to blastocysts, and we observed robust depletion of either Nanog or SMARCA4. And then we performed CUT&RUN for Nanog. And we found that embryos depleted of the ATPase reduced Nanog occupancy dramatically, and in fact to similar levels as Nanog knockdown itself. And similar to the previous studies I showed you in mouse embryonic stem cells, we found by CUT&RUN also that BAF is not required for Nanog localization in these ES cells. And so these data demonstrate that BAF is required for appropriate Nanog localization during development, but not in these cultured cell lines. And so there's really important differences between the developing embryo and cells cultured from the embryo. So overall, the conclusions here are that Nanog is significantly more dependent on ES-BAF for chromatin association in vivo than in these embryonic stem cells. So I again threw these slides in at the last minute because they told me I had a little bit of extra time. And I wanted to talk to you about really preliminary data we have about applying CUT&RUN to different tissue samples and stuff, because I thought that might be of interest to this community. So our collaborator, Yang Liu, is really interested in colon cancer. And we can use the APC-Min mouse as a model for human colon cancer. I'm not going to go into the biology here, but it's been used since 1990 to mimic human colon cancer. And so Yang's group is a microscopy group, and they altered a super resolution microscopy technique called STORM and optimized this to pathological samples. And so this is something, these formalin fixed samples that you take from patients, these super resolution microscopy couldn't be applied to them previously. And so Yang's group was able to optimize STORM, and they termed this PathStorm. And through this application, what Yang demonstrated was that there's a decrease in heterochromatin formation during cancer progression. And this had been observed in the APC-Min mice by H&E staining at 12 weeks. But by doing the super resolution and staining for K9-trimethyl, she can actually observe the decompaction at just 6 weeks. So 50% less time to diagnosis here, which is really exciting and has really important implications, of course. And so to complement this work and to further advance it, thinking about what genomic locations have altered K9-trimethyl binding, we were able to perform CUT&RUN on these APC-Min tumor samples and found a similar reduction in K9-trimethyl, as well as an increase in K4-trimethyl. And of course, then we assessed what were the sites that were, in this case shown here, losing the K9-trimethyl peak in the APC-Min mice at either 6 weeks or 12 weeks. And so it's really great that we could complement the microscopy studies and also identify these genomic locations, because now we have a couple ideas of what changes and which locations are decompacted first during tumorigenesis. The other kind of neat application we have is thinking about applying this to parasites. So Toxoplasma gondii are parasites that lead to toxoplasmosis, and it's considered to be a leading cause of death. I think the leading cause of death attributed to foodborne illness in the United States. So many humans carry the Toxoplasma parasite, and few have actual symptoms because the immune system actually keeps this in check. But women newly infected, either shortly before or during pregnancy, or anyone with a that's immunocompromised, has some severe consequences. And so toxo has a really complicated life cycle. Again, I don't want to go too much into detail here, but when they propagate, they're in their definitive host, which is cats, and then they can actually infect a whole bunch of intermediate hosts, like us or birds. And they can't actually propagate in these intermediate hosts, but rather they become encysted in these bradiozoites, and they can infect mammals in that way. So within the intermediate host, they can't propagate, which means there's really low cell numbers in mammals. But something that, of course, everyone's interested in is what's happening to the epigenome of parasites when they're in different host situations, and how is this affecting the parasite's ability to infect, or many different questions that you can think about. So we decided, because CHIP has been near impossible to apply to toxo in the past, we paired with one of our colleagues here to ask what happens to the parasite's epigenomic landscape in mammalian development. And we're just at the beginning stages of this, but we have been able to optimize CUT&RUN to apply it to these encysted toxo parasites, which is really exciting. So here I'm just showing you from the mammalian host, we recovered the bradiozoites and performed CUT&RUN for K4 trimethyl. These aren't actually over TSSs. I apologize, that's a mistake. But we do see K4 trimethyl localization above the control, the negative control. So then the last kind of random bit of data I want to show you is that we're looking at another type of important disorder, which is this age-related macular degeneration, or AMD, which is the leading cause of visual impairment among the aging population and affects about 10 million people here in the US. AMD, or this macular degeneration, is actually characterized by dysfunction of these RPE, or retinal pigment epithelium. And this is a very small population of cells that surrounds the retina. And so this is a really thin layer of epithelium that overlays the photosensor layer of the retina and plays a really important role in retinal health and homeostasis. And so the transcriptional regulation of RPE has been really well studied. We've been really limited in knowledge of how chromatin organization contributes to RPE function in vivo because of the very few cell numbers we have for RPE in the eye. And so we're working with one of our colleagues in the ophthalmology department here, and we've performed CUT&RUN in either the retina or the RPE. And there is previous ChIP-seq data for the retina, so we're able to compare our data to those. And we do see some of the same peaks or new peaks appearing in RPE. And these data are really under-sequenced, but we're able to get nice profiling in RPE cells. And so we're really excited to continue asking what happens to the epigenome in RPEs during age-related macular degeneration. Okay, great. So we're able to apply CUT&RUN to all these different tissue types, and I didn't have time to show you probably the 10 other tissue types we've tried. And now we can really think about applying this to the most rare and limited biomedical samples. But I do want to switch gears to kind of a more basic side of this, which is thinking about profiling transcription factors in single cells. So due to the high resolution data we were able to obtain for transcription factors in such low cell numbers, we wanted to ask if the technology could be applied to single cells. Of course, this is important because we can learn about the heterogeneity of samples and using these single cell studies. So to that end, we took a population of embryonic stem cells, flow sorted them into single cell per well, and processed each well independently using CUT&RUN. And we assayed 120 single cells for CTCF, shown here, and saw robust enrichment relative to the 47 we assayed using no primary antibody. So these are one-dimensional heat maps. I'm not sure if everyone's used to these, but this is just occupancy shown over all sites, much like a metaplot. But then each row is an individual cell. So you're able to see both the occupancy and the data for single cells. And hopefully you can appreciate that the enrichment for the 120 CTCF targeted single cells is much higher relative to the no primary antibody control. And so now I just want to highlight 12 of these single cells and show you all the data for these. So this is just traditional heat map showing you localization over the 30,000 locations for CTCF that were previously established by CHIP. And you can see again for these 8 that were probed for CTCF versus the no primary antibody control, we see robust enrichment for CTCF. Again, CTCF is a really robust epitope, and so we wanted to see if we could apply single cell transcription factor profiling to other factors. And based on our research interests, we chose to examine SOX2 and Nanog occupancy at the single cell level. We also had reason to know that these antibodies were quite good. So again, here are these one-dimensional heat maps for 26 single cells assayed for SOX2 or Nanog, or some with the no primary antibody control. And again, hopefully you can appreciate that we do get robust enrichment above the negative control background. And probably my favorite data from these experiments is that when we examined a biologically relevant location, which is just upstream of the OCT4 gene, PAL5F1, there are these 3 really important distal enhancers. And SOX2 and Nanog have previously been shown to bind to these enhancer regions. And so here, each row of this browser is the read density for each of the single cells. And you can see just about every single cell profile for SOX2 or Nanog has read density and localization of these factors over these enhancers. So I think it's pretty exciting that we can get robust enrichment of transcription factors from individual cells at these biologically important locations. I'm actually going to skip this slide. So now, shortly after our experiment, Steve Penikoff's group and Keiji Zhao's lab developed wonderful single cell profiling for histomodifications using TN5-based technologies. And they were able to optimize these for high throughput processing for the histomodifications because of TN5 biology and being able to pop that adapter on so nicely. However, as many of us probably know, TN5 isn't suited well for transcription factor profiling studies. And therefore, we think there's a really important application for CUT&RUN and our ability to profile in single cells for some of the transcription factors. And so in light of this, we're trying to really systematically optimize the protocol to tailor it to high throughput transcription factor profiling. And so we're really walking through every aspect of the protocol and trying to incorporate better quantification and optimization for single cell transcription factors and non-DNA binding proteins. And although I don't have time to show you all the data and analyses, we have been performing these optimizations on 500 cells approximately, and have found ways to streamline many aspects of the protocol and get nice data still. So we've also been able to find an optimal way to examine non-direct DNA binding protein or large complexes, like chromatin remodeling complexes like BAF, where we can get far better enrichment using an optimized salt protocol compared to our original CUT&RUN technology, which is wonderful. Okay, so then the last time I'm going to switch gears on you is thinking about exploring the opportunity to pair transcriptomic data with CUT&RUN. So in CUT&RUN, you can either bind cells or isolated nuclei to these lectin-coated concanavalin A beads. And in the case when you release nuclei, the way that we do these experiments, the cytoplasmic fraction is actually discarded. So we figured since many RNAs are exported from the nucleus to the cytoplasm, we could assess transcript levels from this discarded fraction from CUT&RUN. And in fact, we actually already showed that this was possible. So in our previous blastocyst studies, we harvested the nuclei for CUT&RUN, and we used the cytoplasmic fraction for RT-qPCR to quantify depletion efficiency. But we wanted to take this further and asked if we could perform robust RNA sequencing from the cytoplasmic fraction. So what we are doing here is taking a population of cells, performing cut and run as normal, and rather than discarding the cytoplasmic fraction, isolating the RNA and performing RNA-seq. And we think this paired experiment could be important because for many samples, there's a really low amount of material, so you maybe can't get another sample to perform both RNA-seq and cut and run. And also we think performing the experiments in parallel from the same population rather than from different this population harvested at different times could decrease variability between samples. So to test this first, we performed RNA-seq on a titration of cells from 500,000 down to 50 and performed cut and run on the same titration. I'm not going to show you the cut and run data because I basically showed you what that looks like already. And so let's just dive into the RNA-seq data. So here's a genome browser track over a really long gene, SMARCA4. And up top are RNA-seq for the cytoplasmic fraction from 500,000 down to 50 cells, or just whole cells. So total RNA-seq, 500,000 down to 50 cells. And hopefully from this genome browser track, you can see we get really good coverage over the exons, which are these little bars at the bottom. And of course, the lines are the introns. Looking at kind of a less complicated gene, or smaller gene, I guess, not less complicated. We also get really nice profiling from 500,000 down to 50 cells from either cells, which millions of people have done, or from the cytoplasmic fraction that's discarded during cut and run. And so if we look at the RNA-seq coverage over all TSSs in either 500,000 cells or 50 cells, we get, again, oh, it hasn't advanced. Sorry. Again, qualitatively, fairly nice data here as well, where perhaps as expected, the 50 cell data is a little bit more sparse. So we're getting qualitatively between the cytoplasmic fraction that's discarded in RNA-seq compared to traditional whole cell RNA-seq, or all the RNAs, we get really nice mRNA expression profiles. And so finally, here's, my slides are really slow advancing, I'm sorry. Here is the genome-wide coverage correlation across all the samples using just Pearson correlation. So again, this is correlating the 500,000 down to 50 for the traditional RNA-seq or just the RNA-seq performed on the discarded cytoplasmic fraction. And we think that these correlate pretty well. And there's no worse or better correlation from the cytoplasmic or the traditional RNA-seq. So we're pretty excited about the possibility this experimental pairing offers to us and everyone else. And so with that, I'd like to conclude and just review what we covered. So we were able to profile transcription factors in very low cell numbers using cut and run. Furthermore, application of cut and run to embryos demonstrated a role for BAF in regulating nanog localization, which had not been observed in cultured cells. We were also able to profile transcription factors in single cells, which could permit insight into the heterogeneity and factor localization across cells. And finally, we can perform fairly robust RNA-seq from traditionally discarded cytoplasmic fraction and therefore pair RNA-seq with cut and run. And with that, I'd like to thank our fabulous group, which is composed of an incredible set of talented people. Santana is the one in the lab who's been working out all these tweaks with with cut and run to get that going for us. And I started the published work when I was a postdoc with Tom Fazio, who's absolutely incredible, and finished the work in my own lab here at Pitt. Anna Boscovich, the talented embryologist I told you about, postdoc with Ollie Rando, and she was my partner for the embryology work. And so I'd also like to thank some of my funded sources and all of you for listening. So happy to take questions.
So thank you so much, Sarah. That was a fantastic seminar. A reminder, people, you can either type your questions into the Q&A box or you can raise your hand and we will unmute you. It looks like we have a couple questions here in the Q&A. We'll start with that. Sandy Pan from the Hainer lab is asking, hi, Sarah, thanks for the wonderful talk. Is Nanog expression in ES cells much higher than in blastocytes? If yes, do you think that explains the lesser dependence on Brg1 for Nanog binding in the embryonic stem cells?
Thanks for the question. Nice to see you too. Well, not see you, sorry. Yeah, so the expression level, I mean, it's hard to compare across different cell types, but it's high in both. So Nanog is actually expressed in the trophectoderm as well. And I don't know if you caught from the RT-qPCR, there's actually an increase when you knock down SMARCA4. There's an increase in Nanog expression, and we think that's actually the trophectoderm level is increasing. So there is a contribution from the trophectoderm that isn't present in ES cells. But overall, no, I think it's a developmental thing. I think you've captured what's happening. You've kind of frozen things in the ES cell state. You're no longer reliant on these remodelers that you are reliant on during actual development.
That's great. Okay, and then Irene Kaplow asks, in bulk high throughput sequencing data, it is common for transcription factor to have low expression of cell type, but have thousands of ChIP-seq peaks. In the single cell data, do you see transcription factors with low expression having many binding sites, or do you see transcription factors having high expression of small subset of cells having many binding sites in only those cells?
Yeah, so the single cell data that I showed you is the only single cell transcription factor profiling we've done. So literally those cells. So there's not tons of data. We don't have this high throughput like the big labs have for the histone modification. So we only have a few data sets here. Now, CTCF is actually a factor that's really lowly expressed in the expression level, and this is an example of one that binds many locations. And so we did get better profiling of CTCF than SOX2 and Nanog. I think that's more an epitope thing than it is due to the expression level or anything like that. But CTCF is a great example of a lowly expressed gene that you can get lots of nice binding peaks for.
That's great. Okay, and then Andrew Field has his hand raised. So Andrew, I'm just going to allow you to talk. If you can unmute yourself, you can ask your question.
All right. Hi, Sarah, and thanks for the wonderful talk. I was just wondering, you showed a beautiful example of the OCT4 enhancer being co-occupied by SOX2 and Nanog. And I was wondering if there are any cases where you see where there's some sites that are only occupied in some subfraction of the cells, and if there's any interesting insights from that.
Yeah, I mean, it's a great question, and I hate to give you a really crap answer, but because we only have a few cells, it's really hard to do that analysis. So, you know, I only profiled, because it's low throughput, 26 cells for SOX2 and 26 for Nanog. So it's really low amount of data, but there's definitely variability in where you're seeing binding in each individual cell. Now, that being said, this slide I kind of skipped over really quickly because I realized I went too long. It is the peaks that have higher ChIP-seq peak enrichment. You tend to get them recovered more in the single cells, which is logical. So the peaks that you see more enrichment in high cell ChIP-seq, you get more coverage, and more single cells have that coverage. So that's kind of the general assessment I can give you. We can't dive too much into those analyses because we just don't have enough data.
Gotcha, but I'm curious if there are any strong signals that you see in ChIP-seq and that are maybe only in, I know you only have a few cells, but if they're only in half the cells or something, that could be potentially informative. But thank you.
Well, yeah, I mean, even the that example, that browser track I showed you that I can't figure out how to get back to. I'm on my phone, I'm sorry. The promoter in ChIP-seq data has SOX2 Nanog, and not that many of the single cells actually showed that enrichment. It was the enhancer really had the tracks. If I can get back there, I can show that to you. I don't know how to do it, I'm sorry. But yeah, so there are definitely examples like that.
All right, very
I haven't
cool.
quantified it though, yeah.
Yeah, and Peter Sarkis actually had a question on, I think, on that exact point, asking in the browser window that you showed for the single cell CUT&RUN profiling, he was asking about if the peaks from the bulk and the single cell don't always match up exactly. Do you have an explanation for this?
Yeah, I mean, there's huge heterogeneity in single cells. We've seen this in RNA-seq and attack-seq, in single-cell attack-seq too. It doesn't always line up that the locations, and single-cell Hi-C is probably famous for this now. You don't always get the same results from it. For example, if I aggregate 50 cells from the CTCF single cell data, I don't get nearly the same math that I get from 50 cell CTCF profiling, for example. So it's spotty. We get poorer data in single cells, which is to be expected, but you're definitely not getting the same coverage for some reason, which is very interesting.
Yeah. Okay, and then Manny Adam has asked, what was the depth and sequencing do you manage to do with the cytoplasmic RNA-seq? The data looks very robust and adds exciting work.
Yeah, thanks. Can I remember that off the top of my head? I should. I did all that analysis. I honestly can't remember. I'm sorry. But it was very robust. That's why I showed you the correlation. We got really nice coverage. And now we only focused on the mRNAs. As you can imagine, we don't really get much other than mRNA signal there. But yeah, I'm sorry. I can get you those numbers later.
Yeah. And then Mano Saxini asks, hi Sarah, excellent talk. Did you check for OCT4 occupancy as colocalization with BAF and ES cells?
Okay, so we have done some BAF cut-and-run profiling, but we only just really recently optimized it to get that much nicer profiling. And so we actually haven't compared our cut-and-run data for OCT4 and BAF to look at co-occupancy, but that's something we could certainly do. Thank you.
Yeah, and then Amelia Garnier asks, did you try to dissect blastocysts in order to analyze separately the ICM and TE with cut-and-run? If yes, did it work?
Yeah, we wanted to do that, and then I left my postdoc and had no embryologist to do it with me. So I am not an embryologist. I can't work with embryos at all. I now have some partners here that we're working with, but I wanted to ask that exact same question and didn't have the opportunity, but I'll keep you updated.
Awesome. And let me see, I think that's all the questions. Oh, Jingzhao Zhang asked for quantification and comparison of different samples. Is there any spike in strategy to allow better comparison?
Yeah, so for cut-and-run, the Henikoff group has really robustly described this. So you can spike in a heterologous DNA at one point during the end step of the cut-and-run and before you do the library build. Or actually, if you purify the protein A or protein AG emanase on your own, you often get some contamination from the E. coli. And so you can use that those contaminating reads from the E. coli as a spike. And that was Mike Mears' paper, E-Life, maybe, I don't remember. Mike Mears from the Henikoff group. I can't keep track of all their papers.
Yeah, they're prolific. Okay, great. That's fantastic. Thank you so much, Sarah. I think we'll wrap it up there.